Hello and welcome to this series of videos on anti-infectives. And this is part 1 of 3. My name is Dr. Bill Deal-Jones. I'm a professor in the Faculty of Health Disciplines at Athabasca University. So in this series of 3 videos, we'll begin with part 1. And part 1 I'm calling Microbiology 101, which is a review of basic bacteriology. Part 2 is on bacterial virulence factors, and part 3 is a longer video on antibiotic mechanisms of action and classes of antibiotics. So our agenda for part 1, or Microbiology 101, is as follows. We'll begin talking about the anatomy of a bacterium. What are the basic components? Then we'll talk about gram staining, which leads nicely to a discussion of how bacteria are classified. So let's look at the basic anatomy of a bacterium. Bacteria are prokaryotes, while we are members of the class of eukaryotes. Prokaryotes do not have internal cell membranes. And so in this case, you'll see that the genomic DNA sits around free in the cytoplasm of the bacterium without any nuclear membrane. As well, prokaryotes tend to have plasmid DNA, which are small rings of DNA which contain luxury genes. And we'll talk more about these later. There is a plasma membrane in both pro and eukaryotes, but bacteria have got a cell wall. At least most bacteria do. I'll give for you some exceptions. There is also, in some bacteria, a capsular slime layer. Other features of bacteria, there may or may not be flagella, and there may or may not be pili or fimbria, which are extensions which promote attachment and reproduction. So more on bacterial cell walls. We use gram staining to differentiate between 2 types of bacterial cell walls. And gram staining is a multi-step, multi-dye process, which leads us to distinguish between gram-positive and gram-negative bacteria. Gram-positive bacteria have got a cell wall with a thick peptidoglycan layer. And this is what enables gram-positive bacteria to retain more of this purple dye called crystal violet. Gram-negative bacteria, however, have got a cell wall with a thinner peptidoglycan layer, and in this case, the bacteria cannot retain the crystal violet, but do pick up a pinkish dye called safranin. And this shows a field of bacteria, most of which are showing a deep purple color, making them gram-positive. Now, let's take a deeper look at gram-positive and gram-negative cell walls. The gram-positive cell wall, I mentioned, contains a thicker peptidoglycan layer. And in the diagram at the upper right, you can see the top layer of peptidoglycans linked by teichoic acids. In gram-negative cell wall, which is arguably more complex, there's a thin peptidoglycan layer, and you see it more buried in the middle, the bottom picture. And it has an upper layer of lipoprotein and lipopolysaccharide. And it turns out this lipopolysaccharide is pretty important, and we'll talk about it later. So I mentioned there are some exceptions to having a cell wall, and these are the major exceptions. Mycoplasma bacteria, as well as chlamydophila bacteria and Legionella, all lack a cell wall. And some, like the chlamydophila, are intracellular parasites, that is, they live inside most cells. Now, let's subdivide gram-positive and gram-negative bacteria according to cell shape. And while I'll go through each of these different types of bacteria in turn, first thing to point out is that gram-positive bacteria come as either bacilli or rod-shaped, cocci or round-shaped, or branching filaments. And what's important to remember is that within each group, we can further divide these based on different biochemical properties. So again, bacilli, cocci, and branching filaments are the main types of gram-positive bacteria. So turning first to the gram-positive cocci, these are all aerobes. That means they use oxygen for life. And we can categorize them as either catalase negative or catalase positive. And the streptococci are basically bacteria, cocci, which grow only in chains and have two main groups, A or B, which cause strep infections. Staphylococci, which grow in clusters, are very commonly found on the skin and in the respiratory tract, and at least 20 to 30% of humans are carriers. And it's staphylococcal infections, which are the most common cause of skin infections. With respect to bacilli, we can define them as either aerobes or anaerobes, and the anaerobes do not need oxygen for survival. And one of the aerobes is Listeria, named after Dr. Joseph Lister, who was one of the first to use in clinical practice antiseptics. A major human pathogen is Listeria monocytogenes, and this causes gastroenteritis in humans. C. difficile is an anaerobe which lives in the gut, and it can produce toxins that cause diarrhea. They're also very tough because they form endospores. Now, how about the filaments? Some examples of filaments are as follows: the Nocardia and Actinomyces. Nocardia are aerobes, Actinomyces are anaerobes. Nocardia can often produce, they're very rare, but lung, brain, or skin infections, whereas in Actinomyces, they're fairly uncommon. They may be secondary to periodontal surgery, and they slowly grow in areas of the face and neck. Gram-negative bacteria, based on shape, can be categorized as follows. We have, again, bacilli, but also diplococci, which are clusters of two cocci. Coccobacilli, which are mixed between bacilli and cocci. Vibrio, which are bent-shaped, and spirochetes, which are, of course, spiral. And again, we'll talk about each group in particular. So the gram-negative bacilli, or rod-shaped bacteria, are classified as to whether or not they ferment lactose. Shigella is a non-fermenter, and it's closely related to E. coli, and it is a cause of dysentery. Pseudomonas is found in soil and can cause skin rashes, so they're both non-fermenters. Enterobacter and E. coli are ones to watch because they can cause infections in the urinary tract, and Enterobacter also in respiratory tract. E. coli, or Escherichia coli, has many strains. Most are harmless, but a few, like the one shown here, is a cause of hemolytic uremic syndrome. Gram-negative diplococci include the following: Neisseria gonorrhoeae and Neisseria meningitidis. And gonorrhea causes urinary tract infections, whereas Neisseria meningitidis is a common etiology of bacterial meningitis. The gram-negative coccobacilli include Haemophilus influenza and Bordetella tussis. Haemophilus influenza primarily causes illness in babies and kids, has nothing to do with flu, however, it's not a virus, and it's a possible cause of bacterial meningitis. Bordetella pertussis, you should all recognize as being one of the major etiologies of whooping cough. The gram-negative Vibrio bacteria are bent, roughly speaking, and Vibrio cholera is one member of this group. and it produces a toxin that binds to G protein, upsets fluid balance across the gut, and can cause severe diarrhea. Helicobacter pylori inhabits the stomach and is associated with peptic ulcers. Finally, the last group of gram-negative bacteria include the spirochetes. These are spiral-shaped, and there are a few exemplars of this class, but the main one to remember is Treponema pallidum, which is the causal organism of syphilis. So I hope this has been informative and a little bit enjoyable. This is the end of part 1. Thanks, and stay tuned for parts 2 and 3.